Alright, so let's look at step number 1, transcription. So a second ago you told me that it happens in the nucleus. So this is my location. And then we talked about how you start with DNA. That's my original recipe. And I'm going to end up making mRNA, messenger RNA. Messenger RNA is going to be my ingredients because we compared this to making pancakes. mRNA is kind of like that sugar, that flour, those chocolate chips, everything that you need to actually make the pancakes. If you want to make chocolate chip pancakes. Anybody like chocolate chip pancakes? Raise your hand. Ooh, I'm with you. Love me some chocolate chip pancakes. Anybody ever seen the bacon pancakes? You see them, it's pancake, but then people, like they had a griddle and they put bacon down and they cooked the bacon, but then they poured the pancake batter on top of the bacon whenever it was done. And then you don't have to, you know, take a bite of bacon, take a bite of pancake. It's just all in the pancake. I've never tried it, but some people like it, some people don't. So this is what I start with. This is my end product. So I'm starting in the nucleus. I'm going to take a piece of DNA. Let's make up a random DNA code. Remember, DNA has what 4 nucleotides? Adenine, guanine, thymine, cytosine. So let's just say C, A, G, G, T, A, C, A, G, T. That's my DNA strand. We're going to say that we're going to make mRNA from the 5 to 3' strand, the leading strand. And I'm going to need someone to help me turn this DNA, this template, this recipe, figure out what ingredients I need, figure out what mRNA I need. So who's my enzyme that's going to help me do this? So I'm going to have this little guy, this blobby blob. He's RNA polymerase, RNA poly for short. He's going to come in here and he is going to read my DNA. That's his job. And he's going to match a corresponding nucleotide, a corresponding letter to my DNA strand. So if we think about this, this is going to be my RNA strand that I'm going to build. It's going to be in pink. What nucleotide always goes with C? Where do cars go? In a garage. But think about apples now, that A. Are they going to go in the tree or under the tree? They're going to go under the tree. That's your number 1 trick. If you're sitting there going, ooh, adenine, it used to go with thymine, but I don't remember. Apples go under a tree now. What goes with guanine always? Cytosine. What goes with thymine now? You ask yourself, what always goes in a tree? Apples. Adenine. My apples go where now? Under the tree. Then let's just name the rest of them. So what goes with C? G, U, C, A. So this is what RNA polymerase does. Now, there are kind of 3 underlying steps of transcription. There's initiation. So initiation. B, elongation. We kind of talked about those in the video, but I want to make sure you understand them. And then C, termination. To initiate something means to start it. To elongate something means to build it. And then to terminate means to stop it. So where these things come into play? Initiation, the starting point is whenever RNA polymerase comes and finds the gene, this whole piece right here, this segment of DNA. We're not copying all your DNA, all 6 feet that's inside one cell. We're not going to do that. This is a gene. A gene is a segment of DNA. It's a part of the DNA that codes for a protein. So it's going to find the gene. Once it finds that segment that it wants, it's going to start reading it. Elongation is whenever that RNA polymerase is actually putting in and building the mRNA strand. It's saying, hey, I have a C, I'm going to go get me a G nucleotide and put it on there. There's an A, I need to put a U with it. It's reading, actively reading that DNA and building the mRNA. It's reading each nucleotide and figuring out what goes with that until it reaches the end of the gene, the end of that segment of DNA. Once it finds the end of that gene ended with the nucleotide adenine, it's going to terminate. It's going to stop reading. An RNA polymerase is going to go along its way, and it's going to find another gene it needs. So that's what it's going to do. Now, this is not where transcription kind of ends. Yeah, we terminated. We stopped reading the DNA. We made a strand of mRNA. But we've got to do something to that mRNA so it can leave the nucleus, go to a ribosome, and be made into a chain of amino acids that will later fold up and become a protein. So, do I need to leave this up here for a second? Alright, someone make sure that you can tell me the mRNA strand so I can do something with it. Anybody got that written down? Okay. Yay! Alright. This is after termination. So this is the modification of mRNA so it can leave the nucleus and not get eaten in the cytoplasm. The cytoplasm is a cruel world. It's got digestive enzymes out there and not get eaten. You don't want to get eaten. It'd be bad. So we have our mRNA strand. If someone will read that off to me very quickly. What is it? G
U C C A U G U C A.
Thank you. So, this is my mRNA strand. This is what I just made. I need to modify it. I need to go through some modifications. What does it mean to modify something?
Change.
I need to change it. Very nice. So I'm going to add some things that's going to help this mRNA not get eaten in the cytoplasm. You don't want to get eaten. You don't. It'd be terrible. And then I'd be sad. This is what we're going to do. This is my 5' end. This is my 3' end. At my 5' end, I'm going to add a 5' cap. That's protecting my mRNA at the 5' end. That's going to help it. At the 3' end, I'm going to add a poly A tail, which just means that it is a long series of adenines at the end. That's protecting my mRNA at the 3' end, a poly A tail. So that's at the two ends. Now, there's also going to be something that happens in the middle. So I got my 5' cap, got my poly A tail. There's something we're going to have to do in the middle. It's called splicing. Splicing. Splicing means to cut out. We're going to need an enzyme for this. It's called a spliceosome. Let me make sure that I spell it right. Because if I don't, I will feel terrible forever. So elongation. Yep, I knew it was a C. A spliceosome. That's my enzyme. He's going to go in and he's going to cut out something. What are we going to cut out? We're going to cut out my exons, my coding segments that make a protein, or am I going to cut out my introns, my segments that don't code for a protein? I'm going to cut out introns. They don't code for a protein, so we don't really need them. They have a different job. So let's say that, let's say that an intron in here is CAUG. That's an intron. That's an intron in this gene. I am literally, if I'm a spliceosome, I'm an enzyme, I'm going to come in here and I'm going to cut him out like a pair of scissors. I'm going to come in here and I'm going to erase him. Cut him out, bleep, bleep, gone. And then I'm going to also come in here so I can cut things out. I'm also going to stitch those two exons, my actual segments that code for a protein. I'm going to take them, I'm going to splice them or stitch them together too. So I can splice, I can cut, and I also can stitch together. I cut out that intron, boom, it's gone. I don't need it really, so I get rid of it. And I stitch together my two exons. So once I have done all these modifications, I've done all these changes. I've added a 5' cap, I've added a poly A tail, I've spliced out my intron, I've stitched together my exons. I now can go to the cytoplasm. I have done all my checkings. I have done all my modifications. Now I can go to the cytoplasm. I'm going to go attach to a ribosome, and I'm going to make a chain of amino acids and then fold it up, make a protein. Anybody still writing this, raise your hand. All right, keep going. All right, we've cleared the board. We're going on to step 2, translation. You told me a second ago that this occurs in the cytoplasm at a ribosome, because ribosomes are the actual factory for making amino acid chains. And then a minute ago you told me that we're going to take my mRNA that I just made and I modified, and I'm going to turn it into amino acids. The building blocks for protein. I'm going to take my amino acid chain after translation, and it's going to go to one of two places, depending if it's going to exit the cell or if it's going to stay inside the cell. Because remember, proteins are your workhorses. They either do things in the cell membrane, they're enzymes, they do something. So again, I'm going to have that A, B, and C part. I'm going to have initiation. I'm going to have elongation. And then I'm going to have termination. And they still mean the same thing. To initiate means to start. To elongate means to build. Terminate means we're going to stop. So if we take the mRNA strand that I just made, I know it was GUC. I spliced out my intron, and then, what was my second part? UCA. So at the 5' end, I have a 5' cap. I've moved out of the nucleus through those nuclear pores. At the end, I have a poly A tail. That's at the 3' end. So remember, it's just a bunch of adenines added on as protection so that things don't digest this mRNA strand whenever it goes to the cytoplasm. It's a cruel world out there. So, what is going to happen is that I'm going to have another key player here. It's not an RNA polymerase, it's not a DNA polymerase. Who's my person, my lovely cellular particle that's going to read this mRNA and turn it into amino acids? What is it, Chance? I'm going to need transfer RNA in a minute, but who's going to actually read? It's got two subunits, large subunit, small subunit. Big old blobby blob. Think about parts of a cell. What's the part of the cell that makes protein? Ribosome. Ribosomes are going to initiate translation by finding this 5' cap on my mRNA. It has a large subunit that actually does the reading. It has a small subunit. Ribosome. Large subunit does the reading. Small subunit is kind of like the working space. It holds, like your working space is your desk. That's where your papers sit. That small subunit is where the mRNA is just kind of going to have a seat while the large subunit reads it. And it reads it 3 nucleotides at a time. What are 3 nucleotides at a time called? That is right, codon. It's going to read it 1 codon at a time. It's going to read GUC. And this is where the ribosome needs a buddy. It has a buddy. Anybody? Someone just told me who his buddy is. It's kind of like his gopher. He's going to say, hey, go find this, bring it back to me. Transfer RNA. He's going to say, hey, transfer RNA. tRNA for short. He's actually T-shaped. He has an anticodon on the bottom that matches up to that codon. His anticodon for GUC would be CAG. GUC corresponds to an amino acid. And if I can get my amino acid chart, I'll tell you what it stands for real quick. So, GUC. Where are you at? GUC stands for valine. VAL for short. That ribosome reads GUC. Hey, thymine. Or pardon me. Hey, tRNA. You carry amino acids for me. You have an anticodon of CAG. GUC codes for valine. Go get me a valine, please, and bring it back. He's going to say, okay, boss. Ribosome is his boss. He tells him what to do. 2-tRNA doesn't have anything else to do that day. That's his job. He gets paid. So he's going to say, okay. He's going to come. He's going to sit down. And he is going to deposit his valine. But can that tRNA leave yet? Think about a bunch of people going to the bathroom. Can he just leave? No. He is going to have to wait for someone else to come and sit down. Ribosome's going to move on. He's going to come in here. He is going to read UCA. He's going to tell the tRNA that corresponds with the anticodon AGU. He's going to tell him, hey, I have the codon UCA. That codes for my amino acid called serine. He's going to come. He's going to sit down. And he's going to deposit his amino acid and this tRNA can leave. If this was a longer chain that we're forming, if this was a longer mRNA strand, we'd have a longer chain. This protein is just 2 amino acids long, but that's pretty rare. This amino acid chain, we're going to take it. This is its primary structure. If you remember back to molecules of life, proteins have primary structure, secondary structure, tertiary structure, quaternary structure. This is primary structure. It just kind of looks like a line. But remember, proteins are folded up chains of amino acids. Elongation happens whenever this ribosome is actually walking down the mRNA strand and telling the tRNA, "Ho, go get this amino acid." It codes for this codon. Termination is whenever he gets to the end of the line, usually a stop codon. And this one, I didn't have a stop codon, sorry. And he's going to terminate. That ribosome's going to leave. tRNA, they've already left. They've done their job. They're good to go. And usually this mRNA is going to be recycled back, and you can make more amino acid chains from it. Then we're going to take this amino acid chain and we're going to fold it up to its tertiary structure, and then it will be called a protein. And we're going to do that in just a second with pipe cleaners. Alright, so any questions? Anything you want me to go over again? Okie dokie.